All right, we're going to look at genetics today. We're going to look at how a field full of these purple irises can then have a few white irises in there. How does this happen? What is going on with the genetics here? Let's start with this guy right here named Gregor Mendel. He started out just an ordinary guy, boy growing up on a farm. And then he became a monk, a scientist, a mathematician, and the founder of modern genetics. His background in science and mathematics and his background being a farmer led him to notice this garden pea plant. Garden pea plant is just an ordinary plant. However, it has a lot of characteristics that are either or. So you'll have seed shape. It's either round or wrinkled. Seed color, it's yellow or green. Flower color, purple or white. Pod shape, inflated or constricted. Pod color, yellow or green. And then you have the position of the flowers, axial or terminal, where the flowers are on the plant. Stem height, tall or dwarf. There's all of these different characteristics, and he started noticing patterns in the characteristics of these plants within his garden when he was a monk. And his background in mathematics really helped him to analyze those patterns he was looking at, or he was seeing in his monastery garden. Okay, so he started out, let's look at flower color. He noticed that there were a lot of purple flowers and some white flowers. So what he did was he took the purple flowers and bred them only with other purple flowers. And then he took those purple flowers and bred them only with other purple flowers until all he got in that whole garden there was purple flowers. And he called those true breeding purple flowers. And then he did the same thing with the white. He took white flowers and he bred them with other white flowers. And he took those flowers and bred those with other white flowers until all he got when he bred them together were white flowers, no purple at all in that garden. And he called those true breeding white flowers. So the fact that there are purple and white flowers shows us that there's two different codes being passed from parent to offspring. And those two different codes are called alleles. Alleles are different versions of a code, or of the gene. So right now, we know that we only have purple flowers and purple genes in this garden because that's the only kind that we have now. And in the other garden, we only have white flowers. So we have true breeding purple and true breeding white. And then he took one from each garden. Well, he did this many times. But each time he took one from one garden and one from the white garden, and he bred them together. And that is the F1 generation. It's the first generation hybrid. And even though he took purple and white and put them together, all of those flowers that resulted were purple, all of them. And we know they're hybrids because they're a mix between pure purple and pure white flowers. So then he took these first generations that were all purple, every single one of them purple. And he took two of those and bred them together. And he did this many times. Hold on one second. Whoops. Here, I'll show you that in a minute. I got ahead of myself. Okay, so with this F1 first generation hybrid, because they're all purple and there's no white at all, we know that there must be a dominant allele that's masking this other allele, the recessive allele. And the way we know that is because even though there's at least one version of purple and there's one version of white in each one of these flowers, because they have half of their genes from one parent and half from the other, and yet only the purple parent is showing. That tells you that that is a dominant allele. We're going to give it a capital letter P for purple. And it's masking the recessive allele that we know must be here for the white flower. And we're going to give that a lowercase p. It's a recessive allele. So those are more terms that you need to know, dominant and recessive. Okay, so we have our F1 generation that are all hybrids, and they're all showing the dominant allele of purple. Now, we know that they got one dominant allele and one recessive allele in each one of those. So we can tell what their genotype is, which is going to be one capital P and one lowercase p for each one of these. And we know what the true breeding purple flower was. It's two. It only has two copies, only the purple allele, the only allele here. It never made a white flower. And then this white flower over here has only the lowercase recessive allele because the lowercase p means white. Okay, So when we breed these F1 generations together that had one of each allele, we actually, in the F2 generation, get 3 purple flowers and 1 white flower, so that white showed up again. Mendel noticed this in his garden, and he started wondering, what is going on here? How is this possible? Well, we know for sure that the genotype of that white flower has to be 2 lowercase p's. It has to be 2 recessive alleles, because the recessive allele leads to a white flower. Now, the dominant allele is the one, if there were a dominant allele, would have been a purple flower. So we know each one of these has to have a dominant allele. It has to have a capital P. It has to have a dominant. But we don't know what the second allele is, because it could be big P, little p, and be purple, or it could be 2 big P's, which would be 2 dominant alleles and be purple. So we're not sure what these genotypes are. It's one or the other, but we know it's not. It's not 2 recessive alleles. Now, one thing to point out, notice how we're only using P's. We're not using P and W. Each gene that you're talking about, each trait, only gets 1 letter. So we can only use P here. When we're talking about white, we would do 2 lowercase p's. And we always use the letter for the dominant allele. So we're using P for purple. Okay, so what's going on here? Well, he just keeps noticing this pattern in all of these different traits. When he looks at seeds, he's looking at true breeding smooth seeds. So we got one garden that's only producing these smooth seeds, and true breeding wrinkled seeds. He has a garden with only these wrinkled seeds. And he does the same experiment where he then crosses 2 true breeding seeds, and he gets an F1 generation where all the seeds are smooth seeds. So that tells you that which one of these is going to be dominant, the true breeding smooth or true breeding wrinkled. Well, if the F1 generation produces all smooth seeds, then we know that the dominant allele must be the smooth. So it must be this capital S for smooth seeds. That's going to be our dominant allele. And then when we do our F1, our F2 generation, when we do a cross of these 2 F1s to produce the F2 generation, then we get a 3-to-1 ratio, just like before. We get 3 smooth and 1 wrinkled. Okay, so if we look at the genotypes here, the genotype for this true breeding smooth seed would be 2 dominant alleles. So this one would be a big S, big S. And true breeding wrinkled seed would be 2 recessive alleles, little s, little s. We cross them together, and each one of these F1 generation hybrids are going to be 1 big S, 1 little s, 1 big S, 1 little s. There are vocabulary terms for all of these, which we're going to go through in just a minute. Oh, and also on this F2 generation, if it's a wrinkled seed, we know it has to be 2 little s's, 2 recessive alleles. And I'm going to give you guys practice on these also. Okay, and this is just saying the same thing that we just talked about. But we're going to throw in some vocabulary words. This word phenotype. Phenotype is going to be a description of what the trait physically looks like. So down here, when we're talking about flowers, the phenotype is it purple or is it white? That would be the phenotype. Or with the seeds that we looked at, it would have been wrinkled or smooth. Now the genotype is going to be what genes we're looking at. So phenotype, that PH, you can look at physical phenotype, and genotype, you can look at gene. So what are the genes, or what are the alleles that we're looking at? Down here in the true breeding parents, we have true breeding purple. Purple would be the phenotype, and white would be the phenotype of the other parent. The genotypes, we got more vocabulary word for you. In the true breeding parents, they're 2 of the same versions of the code. So they are homozygous. Remember, homo means same? We've talked about that before. When something's homo, it's the same. So 2 of the same versions of the code for a trait. So one of them, this parent right here, is homozygous dominant, meaning it has 2 dominant alleles. Where this flower right here, this white one, is going to be homozygous recessive. It has 2 recessive alleles. And then they produce these F1 hybrids, And all of the hybrids are going to be heterozygous, which means different. They're not the same. Hetero, different. So do two different versions of the code. All F1 hybrids are going to be heterozygous. Now the F2 generation, we know that each one of these purple flowers has at least one dominant allele, but we don't know what the other allele is. And we know that the white flower has two recessive alleles, or homozygous recessive. So you can't say homozygous. You have to say whether it's homozygous dominant or homozygous recessive if you're asked for genotype. You can just say, if you're asked for genotype and you're looking at the F1 generation, you can say it's heterozygous. That's fine, because that's the only thing you can say. Heterozygous means one copy of each. So those are vocab terms that you will need to know. Okay, so Mendel, he had a really strong background in mathematics. And he started looking at these patterns and he thought, man, I think I know a way that can explain these patterns that I'm seeing. And he used something called a Punnett square. This is the word right here, Punnett square. And it explained the patterns of inheritance for both phenotypes, physical, and genotypes, or genes. So what he did was he took the alleles of the parents and put them up at the top. Remember, each individual or each pea plant has half their genes from the mom and half the genes from the dad, or half the genes from one parent, half the genes from the other. So there's two alleles for every trait, and we take one allele of each and put them up at the top. So let's say this is one F1 plant right here. It has two versions of the same gene, the same allele. So we have a dominant allele and a recessive allele, and this is one plant right here. And we're going to cross this one plant with this other plant over here. We're going to write the alleles on the side. So we have an F1 purple flower, F1 generation, heterozygous, and we're going to cross it with another F1 generation heterozygous plant. Okay, we have two alleles. Now, there's many different combinations of offspring that these alleles could make. So if this allele and this allele were to be fertilized, then you would get homozygous dominant offspring. But maybe it's this allele and this allele that fertilize together. Because remember, an individual plant is going to be diploid. So you're going to have two versions. But after meiosis, you're going to produce gametes. So this represents the different gametes that are possible in this individual. You can have a dominant gamete and a recessive gamete. And this is the different gametes that are possible in the other plant that we're going to cross with. You can have a dominant or a recessive. So if these two gametes fertilize, you get a homozygous dominant offspring. If these two gametes fertilize, you get a heterozygous offspring. And we can keep doing this for each one of these different combinations of gametes. We could have this gamete and this gamete. We could have this gamete with this gamete. So you can see you get 3 purple flowers and 1 white flower possibilities. So those are phenotypes, 3 purple to 1 white. The possibilities for genotypes, we have 1 homozygous dominant right here, it'll be purple. We have 2 heterozygotes, and we have 1 homozygous recessive white. And he did the same thing with all those different traits I showed you. So I'm going to go real quick. Here's the seed color. We have yellow seeds, and we have green seeds. So the recessive gamete is going to be a green seed, and the dominant gamete is going to be a yellow seed. And you can see you get the same pattern, 3 yellow to 1 green, 1 to 2 to 1. I'm going to have to stop right here, and I will pick up again with the next PowerPoint, with the next video.